Shri Gurubhyo Namaha children. Today we are going to start with a topic called pollen pistil interaction. Actually, we are learning about gamete transfer, that is pre-fertilization events, which means events taking place before the fertilization in the plant reproduction. Okay, which includes gametogenesis and gamete transfer. Under gamete transfer, first step is pollination. Okay, that pollination is nothing but transfer of pollen grains from the anther to the stigma of the female flower. Okay, now pollination over. Now the pollen grains are deposited on the stigma, and now we are going to see the next step, which is pollen pistil interaction. So this pollen pistil interaction is nothing but once the pollen grain is deposited on the stigma, okay, it has to be accepted by that stigma. Okay, once it is accepted, the pollen grain will start forming a tube, which is called pollen tube. The pollen tube will enter the stigmatic tissue, and it is going down through the style to reach that ovary and entering to the ovary to release their germ cells. So all these events together, we used to call it as pollen pistil interaction. So pollen pistil interaction is the events from pollen deposition. Okay, so they are the events from pollen deposition on the stigma until the entry of the pollen tube into the ovule. So in pollination, pollen is deposited on the stigma, and from this stigma it has to reach the ovary, because ovary contains ovule, which is having a female germ cell. Okay, so all these events which is taking place right from the deposition of the pollen grain on the stigma until the entry of the pollen tube into the ovule, we will call it as pollen pistil interaction. Before going into the pollen pistil interaction in detail, just we'll have a recap of this mature embryo sac. Okay, this is actually the mature ovule will have this embryo sac which is ready for the process of fertilization. Okay, for that fertilization, they need a female gamete. For that, only pollination is already over, and pollen pistil interaction started now. Okay, now, so this is a structure, and this part of this embryo sac will call chalacal end, and this is called micropylar end. This chalacal end is having three cells, which we are calling them as the antipodal cells, and this micropylar end, okay, they have actually three cells, 1, 2 and 3. Two corner cells, which we are calling it as synergids, and the center cell is an important thing, that is egg cell, a female germ cell. Okay, and this synergid is having a finger-like projection here. Can you see this? This is called filiform apparatus. And what is the importance of this filiform apparatus when you see? It is helping in the entry of pollen tube into this ovule. Okay, that is a significant role of this filiform apparatus in this pollen pistil interaction. So this is the structure of our embryo sac, which is present inside the ovary. Okay, so now these are the steps which is there in the pollen pistil interaction. First pollination, then deposition of pollen on the stigma. Once it is deposited, the pollen is hydrated. Okay, it is taking some amount of water present in that stigmatic secretion, and it get enlarged. So as it is enlarging, there will be a process called as pollen germination. We are going to see in detail, and this pollen tube is going towards the ovary by entering into the stigmatic tissue through the style. Then it enters this ovule, which is present inside the ovary. Okay, so we'll see it in detail. Okay, before going into that, you should know something called self-incompatibility. Okay, sometimes the pollen grain, okay, it is from the same plant in the same species. For example, okay, what is it? We'll take a banana. Okay, banana flower is there. So the pollen grains are deposited on the stigma of the female flower of that banana flower. Okay, now sometimes even though it is a banana stigma and it is a pollen grain from that same plant, but they are incapable of fusing. Okay, because of some genetic disorder. Okay, either the male gamete or female gamete is not having the ability to fuse with each another. So this condition we used to call it a sterile condition. Okay, the inability to reproduce. So this self-sterility is called self-incompatibility. So the inability of certain gametes, even from genetically similar plant species, to fuse with each other, we are calling it as a self-incompatibility. This self-incompatibility, it is of two types, that is sporophytic incompatibility and gametophytic incompatibility. If the problem is with the female tissues, we will call it as sporophytic incompatibility. If the problem is with pollen grain, genes of pollen grain, we will call it as a gametophytic incompatibility. So sporophytic incompatibility is due to the genotype of stigmatic tissue. Stigma is not ready to accept its pollen grain. So that condition, sporophytic incompatibility. And if it is due to genotype of pollen, we will call it as a gametophytic incompatibility. So this is about self-incompatibility, which is very important in this pollen pistil interaction. Okay, now we are going to see, okay, what are all the events taking place in this pollen pistil interaction. Okay, so see here, pollen pistil interaction, it is nothing but the pollen recognition by stigma. Okay, if it is incompatible, the stigma will not reject that pollen grain, so that there is no growth of pollen tube. If it is compatible one, it encourages by the pistil to form pollen germination, so that the pollen tube will be formed. So the pollen tube will reach the ovary to release its germ cell. Okay, so before going deep into that, we will see the structure of this pollen grain. Already we have learned this. So this is the pollen grain which is formed, which undergoes mitotic division to form these two cells. Okay, this is a part of a cell, and this small one you can see here, some spindle shaped cell. This is one cell. So this cell we will call it as a tube cell or vegetative cell. Why it is called? Actually, in your book it is given as vegetative cell. Why it is given here as tube cell? When you see, these vegetative cells are useful in the formation of this pollen tube. As it is forming this tube, the vegetative cell is called tube cell. Okay, and this is a generative cell. Okay, this generative cell, it is going to form a next generation. Because of that only it is named as generative cell. This generative cell is responsible for, actually it is going to mitotically divide to produce two male gametes. Okay, so each pollen grain, okay, it is having two male gametes. Those two male gametes are produced by the mitotic division of this generative cell. Okay, this is about the structure of our pollen grain. And the pollen grain, this black color coat is XII, and here and there that black color coat is discontinuous, and those places we will call it as here germ pore. Okay, germ pore. Through this germ pore only the tube is started germinating, and when the tube is started germinating, first the vegetative cell only, it is forming a tube. Because of that only we are calling it as tube cell. Once the tube is formed, the vegetative cell content will enter into the tube. That is, the vegetative cell will come first into the tube, followed by the gametes. That is, generative cell is forming two male gametes, no, those two male gametes. So first the tube nucleus will be there, followed by two male gametes. So this is about the pollen structure. Okay, now on the stigma this pollen is deposited. Okay, then if it is of compatible, okay, it is actually a type of trophic movement in plants. Okay, in 10th standard you have learned about trophism. This is an example for chemotrophism, that is the trophic movement, growth dependent movement of the plant towards chemical. Okay, so here what happens now, in this stigma there is a sugary secretion, okay, which contains some organic acids, so much of sugars. Okay, that chemical is acting as a stimuli for that compatible pollen grain. So this stimuli is recognized by the pollen grain, and the pollen grain started recognizing that stimuli. So because of that only this pollen tube germination will start. Okay, so step by step we'll see what is going to take place in this pollen pistil interaction, how there is a growth of the pollen tube. So as I told you, the pollen grain held by the sticky stigmatic secretion on the stigma. Okay, then next one, the vegetative cell, hydration of pollen grain. The vegetative cell enlarges and it comes out of the pollen grain through the germ pore. I showed you how through the germ pore the pollen tube is coming out. Okay, then the content of the pollen grain moves into the pollen tube. Then the vegetative nucleus, I told you, no, in the previous picture, I told you the vegetative nucleus will be at the tip followed by two male gametes. Okay, then the pollen tube, how this pollen tube is growing towards this ovary. When you see here, it has to grow all the way from here to this ovary. Okay, so in this condition, what happens now? This stigma and style will have some stored food, reserved food. So this pollen grain is secreting some digestive enzymes. Those digestive enzymes digest the complex substance present in the stigma and style, and that digested food is used for the growth of this pollen tube. So using that food only, this pollen tube is growing towards this ovary. Okay, so once it is coming here towards this ovary, see, this is the pollen tube. Pollen tube is having these two male gametes. So on and reaching this ovary, I told you already about this synergic filiform apparatus. This filiform apparatus helping this pollen tube to gain entry into this ovule. So now it is very easy for this pollen tube to enter into this ovule, so that on entering this ovule, it bursts out to release these two male gametes. And these male gametes, see here one male gamete which is going to fuse with our egg cell, and here one male gamete which will be fusing with the polar nuclei. We are going to see that process, double fertilization, in the next topic under fertilization. So this is how the pollen tube started growing, and it results in the release of these germ cells into the ovary. And this overall process, we are calling this as our pollen pistil interaction. So I told you, no, the digested food is used for the growth of the pollen tube, where this pollen tube grows chemotrophically towards the thing, and on entering this, it is releasing those male gametes. One thing to be noted here is 60% of the angiosperms will produce pollen grains in two-celled stage. That is, vegetative cell and generative cell will be there, and all the other process will be taking place. In very few cases, what will happen? Once at the time of dehiscence itself, that is when the anther is releasing the pollen grain itself, the pollen grain will be in three-celled stage. That is, one vegetative cell with two male gametes. Okay, it depends on the plant, but in most of the cases, the generative cell will undergo the mitotic division only when the tube is started developing. That is, when it is in progression, at that time, the generative cell will divide mitotically to produce two male gametes. So on entering this ovule, these two male gametes are discharged into the ovary. So this is called pollen pistil interaction. Thank you, children.